Support Microbe TV. It's time for our annual fundraising drive. We depend on your support to produce high-quality science videos and podcasts, and now is the time to help us. If our programs have appealed to your science interests, or if they have helped you in some way, please record a brief audio or video with your phone and tell us about it. Let us know how we empower your inner scientist. We'll use it for our fundraising efforts. Send it to incubator@microbe.tv. And for more information on how you can give us your support, go to microbe.tv/contribute. From Microbe TV, this is TWiP, This Week in Parasitism, episode number 250, recorded on December 20th, 2024. I'm Vincent Racaniello, and you're listening to the podcast all about parasites. Joining me today from New York, Daniel Griffin.
Hello, everyone.
You must have a STD tie because it's Friday, right?
It's Friday, so I have my short-term disability tie. No, I'm being sexually transmitted disease tie, and it is syphilis.
Curly Q.
Yeah. And usually I don't comment about the weather, but I have to say the weather is quite frightful. It's really cold today. See, my nose is cold from just coming back from walking the dogs through I don't know if you guys have a snowstorm, but we have a bit of a snowstorm out here.
It says here that it's snowing, but I can't tell because the windows are blocked up. It's 2 degrees. And snowing for an hour, so we'll see when I go out after this. Also joining us from Glasgow, Scotland, Christina Nowla.
Hello, good evening, everybody. It's not snowing here, but it's been pretty miserable. I went for a hike this morning, and I was soaked through to the skin.
Does it ever snow there?
Yeah, we do sometimes. Yeah, we do. But because we're on the west coast of Scotland with the Gulf Stream, it is quite warm, so we don't actually get the temperatures that other latitudes that are similar to ours have.
I see.
So no, not much snow. And if we have, everything breaks down.
Right. If you enjoy these programs where you can learn science, we'd love to have your support. In the month of December and January, you can go to parasiteswithoutborders.com and they will match your donation. To help us out. We appreciate your support. Today we have a paper chosen by Daniel, presented by Christina. Go ahead, Christina.
Yes, okay. Daniel, good choice. It was a really interesting read. So the title of this paper is "The Gut Microbiota is Essential for Trichinella spiralis, Evoked Suppression of Colitis." And the authors are Sun, Long, and Yang, and these are joint first authors. And then we have Zhang, Wang, Liao, Qu, and Wang, and they are all from I'm not entirely sure about the Chinese pronunciation of this city, Zhengzhou University in China. So forgive me if I got that wrong. So this is published in PLOS, Neglected Tropical Diseases, and this is an open access journal. So any papers from that journal, you can have a look.
Too bad Dixon isn't here. How Trichinella?
I know.
When I picked this, I was thinking this is right. If we can get anything would get Dixon to, rejoin the fray, like a research article titled "The Gut Microbiota is Essential for Trichinella spiralis, Evokes Suppression of Colitis" would bring him back. But, I don't know.
Yeah, so I think we only recently talked about Trichinella, didn't we? So I don't know if we need to go back into the life cycle or if we just should dive into the paper. What do you think?
We talked about an outbreak in people in Arizona who ate raw bear meat, right?
Yeah, actually, and maybe it serves mention because I know, I listen to the other Microbe TV podcasts. Is that what you call them? YouTube things?
Yes,
productions. I'm going to call them productions, right? Because they're not just podcasts. They're also on YouTube. But so it's this issue that, we always talk about how important it is to cook meat, right? That's how people get trichinella, is by eating undercooked or raw meat. But also the issue we run into, there's certain freeze tolerant species of trichinella. Right. So with bears in particular, you could freeze it for a month. That's not enough. If you don't cook it, you can actually still get it. So, you really got to, and what happens, right? We've talked about you end up with initially a GI syndrome, followed by the muscle invasion and all the issues that go along with that.
I always find it really fascinating that you'd have that whole life cycle really contained in the one host. I always find that quite fascinating. How the adults live in the intestines and then the larvae are produced by the females. That in itself I find quite magical, really, rather than egg production. And then, it continues. I find that a really fascinating parasite. But yeah, I think since we have covered it so recently, we can maybe just go straight into the paper. So if anyone would like to look at the paper and look at the figures, because I think I'm going to talk about some of the figures in a bit of detail, maybe you could just pause your recording and download it following the link that we're providing you. But I think hopefully the take-home messages are also clear if you're not able to download the paper or just don't want to because you're out on a walk with your dog and listening while you're doing that.
I was going to say, I hope David, for the YouTube, maybe he'll even show some of the figures as we discuss them.
Yeah, that's a good idea because it's open access. We can do that, can't we? Yeah. So I thought I'd maybe give a bit of a kind of a background overview before we get started, just to kind of talk. No, I'm not going to talk about Trichinella, but some other bits and pieces. So this paper is talking about colitis on inflammatory bowel disease, and I think maybe we could briefly mention what that is. So there's actually quite a few different inflammatory bowel diseases, but I think Crohn's disease and ulcerative colitis are probably the most common. And so essentially these diseases lead to chronic inflammation in the intestine. So Crohn's disease most commonly affects the last part of the small intestine, the colon, or both. And I think the ulcerative colitis primarily affects the colon and the rectum, and so leads to chronic inflammation and can then also have kind of sequelae, for example, an increased risk of cancer. And it's also, if you know somebody with either of those diseases, they can be really debilitating and they have a really, really unpleasant or difficult effect on those that suffer from it. There's no cure. And Daniel, please correct me on the clinical things here if I make any boo-boos. But I think with anti-inflammatory treatment or treatment with immune suppressants or biologicals, maybe also, or definitely also alongside careful management of a diet and maybe lifestyle can bring improvement. But sometimes we also would need or would consider surgical procedures to maybe remove disease.
You could probably enlist me for the clinical. So
please.
I think that was a great overview. And the two major inflammatory bowel diseases that we discuss are the Crohn's disease and the ulcerative colitis. The Crohn's, can involve small and large intestine. Ulcerative colitis restricted to the colon. So as you can imagine, the surgical treatment could be you remove the colon, and that's the end of ulcerative colitis. But now you've removed the colon. A gentleman in the hospital today who actually he's long suffered with ulcerative colitis, and actually probably a combination of a little bit of a flare leading to a septic picture. He just got out of the ICU. He's on the floor now. And I mean, I don't mean like he's laying on the floor. I mean, he's on the general medicine floor. I just thinking about how that sounds.
Didn't know there was a scarcity of beds.
Yes, exactly. But exactly. So sometimes we'll use even some, I guess, more topical, milder things, salamine, stuff like that. But also, this is an area of medicine that was been revolutionized by targeting TNF alpha. This gentleman was funny. He's not as young as a person might be, but he was wondering, why hasn't my GI doctor put me on these medicines? I watch the commercials on TV and I see these people getting up and dancing. And, if I got on one of those medicines, maybe I'd be doing so well, I'd be up and dancing. And I had to explain that none of these medicines will actually teach you how to dance, but, maybe they oversell them. But no, that's exactly it. This can be actually a really devastating disease. We think of, some loose stools, this or that. But no, people with different forms of colitis, it can be life-changing. It can be debilitating. You can end up with inflammation leading to fistulas, connections from the bowel to the surface. You can end up requiring surgery. So colitis is a major issue. And as we're going to learn about here, maybe there are some insights that we're going to get.
So I think the incidence has really increased over the last 50 years or so, and particularly in the Western world, but also in other parts of the world. It's really a global disease now, but the prevalence really varies quite considerably geographically. And I think also kind of the incidence has been increasing. Actually, the burden of disease is maybe diminishing where and when treatment or improved treatment and management is available. So, for example, I did read a review article that was published in 2023 in the Lancet. So they stated that kind of the burden of disease is reduced. For example, years of life lost or disability-adjusted life years have improved when we compare the data from 2019 with maybe 20 years earlier or even more in 1990. So I think, if these management options are available, maybe, I suppose that is a good thing, but this will not be accessible to many people. I would not think. I guess it depends on where you live, as with everything.
Yeah, it's really interesting, right? Because and they do a nice job in the introduction, right, of laying a lot of this out. But really, since the middle of the 20th century, the incidence of the inflammatory, as opposed to irritable, the inflammatory bowel disease has really increased in, they say Western world, but I'm going to say in industrialized countries.
Yeah.
And what's actually happened, this is maybe this kind of insight that they're working on, is that you take some of these lower and middle income countries, and as they become newly industrialized, you actually see a rising prevalence. So there's been this discussion for a while about maybe getting rid of the helminths, maybe changes in the microbiota associated with changes in diet and other things, maybe driving these increased incidence numbers that we're seeing.
And that's actually a really nice segue into the thing that I wanted to say next. So there is epidemiological evidence that helminth infection, particularly in children, are protective against development of inflammatory disease. There's also experimental animals in using mouse models primarily that infection with some species can improve the inflammatory reaction in inflammatory bowel disease. So, for example, Schistosoma mansemi, a mouse helminth or roundworm with an unpronounceable name, Heligomosomoides polygyrus, and also the rat tapeworm, Hymenalepis diminuta. So that there's some evidence for that in animals. There's also some evidence from human clinical trials which were conducted with ova of Trichuris suis, which is the pig version of the whipworm, but actually the evidence is not really as strong as it probably could be, because the numbers in these clinical trials were relatively low. So there's a meta-analysis that was conducted in 2018 that kind of looked at the effectiveness of Trichinella suis egg therapy, and the studies were included. They were only randomized, double-blind, placebo-controlled trials, so of potentially very high quality in a disease background of ulcerative colitis, but also Crohn's. And there is some clinical remission and clinical response observed in all those trials, but there seems to be not a statistical benefit for inflammatory bowel disease patients. And I think that potentially could be a result of the small sample sizes in those clinical trials. And I think we probably need a lot more evidence and definitely much larger sample sizes to increase the power of those clinical trials, I suppose. And then the third kind of player in that paper is really the gut microbiota. So we've looked at inflammatory bowel disease, and then we kind of talked a bit about the effect that helminth might have. And the third player in this paper is really the gut microbiota. And that's really considered a crucial environmental factor for IBD. And there's accumulating evidence that gut microbiota is a key factor in modulating the host immune system, which can predispose to autoimmune disease and including inflammatory bowel disease. So some species have a protective effect by upregulating IL-10, which is an anti-inflammatory cytokine, and we'll get back to that again later. And there's also potentially downregulation of cytokines that do increase inflammation. And then there's other species of bacteria that have shown to induce an expansion of T regulatory cells, which again will be mitigating inflammation. And then if you bring those two together, obviously many of those helminth that we talk about and the gut microbiome, they share their living environment. And so, that coexistence can induce changes in the composition of the host gut microbiome and maybe promote bacterial invasion from host intestinal epithelium surface to submucosa. So, So if we now kind of bring that all together. So the study that we're looking at looks at the potential influence and the mechanisms of helminth infection, which induces a change in the gut microbiome on the development of inflammatory bowel disease. So the study uses Trichinella spiralis as a model, and that's based on previous studies that these authors have conducted, and also based on studies that shown that other helminth potentially have an effect on the severity of irritable, not of irritable, I'm mixing that up, of inflammatory bowel disease. Very important not to mix those two up. It is 10:30, well, here in Glasgow, so forgive me, but definitely not mix that up. So, for example, they've previously shown that chronic infection with Trichinella can mediate protection against allergic asthma, influenza, respiratory syncytial virus associated pathologies in mice. And so there's also evidence in mice that showed that chronic infection also kind of attenuates the damage caused to the colon by various mechanisms. And this includes also colitis that has been induced in those mice, which we'll come back to in a moment.
I have to say, this is a very, we haven't even got into the data yet, but all this sort of background is fascinating. This idea that your, parasites, gut microbiome, not only that they may play a role in mediating autoimmune disease, but when you get sick, right? When you get a pseudomonal pneumonia, when you get the flu, when you get RSV, maybe part of the severity, as we're sort of seeing this background, has to do with the microbiome that you're living with when you get this infection. So really fascinating, sort of larger picture to all this.
So I don't know, do you want me to give a short take-home message of the paper, or shall we just power through it and then summarize at the end?
Should we go through the data and then we'll sum it all up?
Yeah. So I just wanted to briefly some of the methods as well. So I think just the animal models that were used was an inbred laboratory mouse that is often used as model of human disease. The mice were generally, for all the experiments, they were fed and watered on sterile food and water only, which I think is quite important because we are looking at the gut microbiome. And I think I'm going to describe some of the experimental procedures a little bit, so it's a bit easier to then understand the results. So these animals, so in these mice, you can actually induce ulcerative colitis, or something that kind of simulates ulcerative colitis in humans. You can induce that with dextrane. Oh, I've just lost the word of it. Dextran sodium sulfite, DSS. And so if you do administer that to the mice, it causes human ulcerative colitis-like pathology because it's toxic to colonic epithelial cells, which then results in a compromised mucosal barrier function, which is something that you see in ulcerative colitis. So I want to tell you how they infected the mice first. So for this, actually, it's quite interesting. So they would have to obtain Trichinella larvae. So Dixon, if we had Dixon, he could tell us whether we can grow them in culture. But I think probably not. So essentially, they had to harvest the Trichinella larvae from domestic pigs. They harvested pig muscle, digested it with pepsin and hydrochloride solution so that the larvae would be released, and then they force-fed the mice 100 larvae each for all the experiments, with control mice being fed just saline solution. And then they were left to live their life for 35 days, hopefully quite happy days. And then the mice were treated with that dextran sulfate, that DSS molecule. And so that would be administered over 7 days because it takes some time to take effect. And then afterwards, the mice would get plain water. And then 3 days after having just normal water, they would be sacrificed and processed for sampling. And there's always multiple groups of mice for the paper for each of the experiments. So, just to, I suppose, add power to that study. So generally, I think it was 3 to 4 mice in each of the groups. And then they used those mice to evaluate the role of gut microbiota in Trichinella ameliorated colitis. And for these experiments, they did also treat animals with a broad spectrum cocktail of antibiotics to actually deplete the gut microbiota, so we can compare mice that have microbiome and some that don't. And then the other experiment that they did with mice, so we have quite a lot of different mice experiments here. They did also look at helminth-induced alterations in the gut microbiome and see if that can have an effect if they were putting mice together that did have Trichinella infection and mice that did not have. Because it is well known that, that the microbiome somehow is shared between people who live together and mice that live together. So that's the kind of experimental animals we have, and they are used for all of the experiments shown in this paper. And then, the kind of testing that they did about the effects, they were looking at histopathology. So they would kind of collect pieces of colon and they would measure tissue cytokines, for example. They would also cut them and make sections to look at them and stain them and to assess them microscopically to look if there's any damage to the epithelium cells or the mucous membranes. And they were looking for inflammatory damage. And then they also measured lymphocytes, IL-10 production. And probably it's about 10 minutes ago, but I mentioned IL-10 already, which has a kind of an anti-inflammatory effect. So they would do that as well, and that would give us information on inflammation. And then finally, they also did gut microbiome analysis of all those mice, and they measured some fecal short-chain fatty acids. So I hope that's sufficiently detailed for anyone who's keen on the methods. It is open access, so you can look up the details yourself if you would like to. But I hope that kind of helps you follow through. So we can now look at the results, shall we?
All right, shall we go to figure 1? Are we ready?
Yes, yeah. So the first result shown here is that antibiotic treatment abrogates, and I had to look that word up as a non-English speaker. I wasn't familiar with that. So negates essentially the suppression of colitis in Trichinella infected mice. Abolishes. Yeah. And I suppose the authors are maybe also not native English speakers. I don't know. It's a word that I've not heard, so I had to look it up. So in other words, if there's no gut microbiome present, Trichinella on its own does not improve the symptoms of colitis. And you can see that really in figure 1. So there's quite a lot of sub-figures in figure 1. And I'm just going to have to scroll to it myself.
Yeah, I did this figure backwards, right? Like I looked at E, which I thought E makes it sort of easy for me to see, and
yes,
so they've got
it is.
histological score, and they start with we don't do anything, and the mice are just fine, right? So no T. spiralis, no antibiotics, no DSS, and everything's good. And then you've got all these variations, right? You've got, so what if we give them the T. spiralis and the antibiotics? What if we, and there's no DSS? What if we just do antibiotics? What if we just do T. spiralis? What if we do all these combinations? And, you sort of see the different variations and what you get.
Yeah, it's actually quite nice. So, for example, some things that you can see are quite obvious, really, for anyone, for example, who has had antibiotics treatment before. That on its own already has a mild inflammatory response on the mice, and I react quite badly. So my gut reacts quite badly to antibiotics. So I think that makes perfect sense. And when colitis is then induced, the mice that were treated with antibiotics had much more severe symptoms and earlier than the mice that were not treated with antibiotics. And all the mice with colitis, they showed visible signs of inflammation. And that is, I think, figure B, actually, I quite liked figure B in that figure because that shows the weight loss, which is often, obviously, if you can't absorb food and if you're in pain and you can't eat food and you're suffering, you're also going to lose weight. So that figure 1B actually shows really quite nice the difference in the weight loss. So you can see that 3 mice groups most affected by weight loss were the mice that had colitis in green, the mice that with colitis and no microbiome that are in purple, and the mice with colitis, Trichinella, and no microbiome in blue. And you can also see the positive effects of Trichinella infection on colitis with the red line, really. So these mice lose less weight than the control mice. They still lose weight, but less weight than the other mice that I mentioned in green, purple, and blue. And actually, the histological findings, I think they're quite difficult to see if you're not histology trained. But by all means, go and have a look.
Yeah, you can see, you can see there, just for those of you that have this in front of you, those of you that don't, if you look closely, what are you actually seeing? So the control is what normal gut mucosa should look like. But then probably the best is if you go, there's ones which are labeled DSS, or then particularly the antibiotics DSS, where you actually see a lot of these goblet cells. You see that the area has become dark because you're getting infiltration of inflammatory cells. You're actually seeing this thickening of the mucosa, right? So that's probably a lot of mucus producing cells. But I think that you already have kind of the takeaway here, right? Is that, the T. spiralis, get that in there, a little bit of inflammation, a little bit of histological score going up. You treat with just antibiotics, mess with the microbiome. You're actually seeing a little bit of colitis. Maybe we should be thinking about that, urgent care and primary care docs, when you're throwing those antibiotics around. So T. spiralis, a little bit of inflammation, antibiotics, a little bit of inflammation. But oh my gosh, the DSS, massive amount of inflammation. And the T. spiralis can actually abrogate, right? Not quite abolish, but can decrease substantially the amount of inflammation. But you throw the antibiotics and that T. spiralis effect is really minimized.
Yeah, it's really not abrogation. It's just an amelioration.
Amelioration, yeah. I think
because
that's
it's
better,
clearly, the histological score, the T. spiralis reduces the pathology, but it's only by 5%,
Yeah,
so.
I suppose I just used the word that the authors used. So
yeah, it's fine.
Please
but
forgive me.
I think they're
we're taking issue with abrogating. I think it diminishes a little bit. It's not, because you're getting some inflammation from the T. spiralis, and you're not getting rid of it all. This is not a cure.
Yeah, it's not a magic pill.
You're not going to be dancing like my patient.
No, not going to be dancing.
You're not going to suddenly know the foxtrot. And,
Well, you can send your patient over to Scotland, and we can teach them some ceilidh dancing, which is always really fun.
Okay.
Right.
He is in his 90s, I shall point out.
All right. Okay.
30s.
Maybe not. Well, all ages do ceilidh dancing, from small kids to very elderly people. Sorry, I've lost my thread now because I was thinking about my favorite ceilidh dance.
I thought this was pretty good. And then, are we ready to jump on to figure 2?
Yeah. So figure 2 shows the next important result, and that really shows that Trichinella infected mice alters the gut microbiome composition, and that's without any antibiotics at all. So the infection changes both the diversity and also the composition of the microbiome. And if you, for example, look at figure 2A, so you got a red and a blue bar in particular. So you can see the gut microbial diversity expressed in operational taxonomic units, and that kind of indicates a complex. So the higher that index is, that unit is, the more complex the community is. So Trichinella infected mice have a less complex microbial community than those that are not. And you can also see that actually the composition, I think figure 2D and 2E, and you really just need to focus on the colors, and particularly in figure 2E, you can see these different colors all kind of illustrate different types of bacteria that you can find in the gut. And if you look at the colors, you can see they are significantly different. So I think that's an interesting result as well. And actually then in the next figure, they're also going to look if that... Daniel, did you want to say something? You look like you wanted to say something.
I was going to say, and maybe Vincent, you can jump in on this. It seems almost a little counterintuitive when I got to this figure, because I, sort of have this idea that the more complex your microbiome, maybe that's better. Like we don't want these, and also I sort of had this idea that maybe Bacteroides is not a microbe I'm so excited about, and maybe I want more of the Firmicutes, but I'm actually seeing the opposite of that, right? We're seeing more Bacteroides, we're seeing less Firmicutes, we're seeing less microbial complexity. First, I was thinking this isn't what I want. So it's just seemed to me, I don't know, counterintuitive. Vincent, any thoughts?
Well, it's hard to know because a lot of these changes may have nothing to do with ulcerative colitis, right? There may be some minor bacteria in here that are the key, and we just are not seeing it. That's the problem with microbiome studies, right? You see these shifts, but you don't really know which ones are important.
I've actually had my microbiome analyzed. And it's not good, so I need to eat better. And they actually also, they didn't look at the virome, but they looked at a few parasites. So that the only parasite that they looked at, this analysis was Blastocystis hominis, which apparently is a beneficial parasite. But that may be something for another paper, another day. So actually in these were mice that did not have colitis. I think that was just to kind of show the effect.
But yeah, interesting.
Yeah, of the Trichinella infection.
Yeah.
Sorry, I've already jumped ahead to the next paper.
No,
so I'm thinking that
we can
my
jump to the
title
next
makes
one.
no sense.
No, we can always jump to the next one because I think, so we sort of see this and I don't know what to make of it. Okay. But then this very interesting, like, so what if we co-house? Like, what if you're going to trigger the colitis in both mice, but some of the mice, they're co-housed. Somehow you're not going to get the Trichinella, right? So that's fine. But is the co-housing, can you basically, by just sharing the microbiome, is that going to protect you? So
it's really quite nice, actually, because this shows that co-housing made actually not much of a difference to the severity of colitis in Trichinella infected mice. But in mice without Trichinella infection, the infection of colitis improved as a result of sharing with the Trichinella infected mice. So essentially the Trichinella effect is transferred via, I don't know, licking each other, the gut microbiome, eating feces. Do mice eat feces? I don't know.
Yeah,
some rodents do. They do.
Yeah.
What's that name?
Coprophagy.
Coprophagy. So, yep. That really demonstrates quite nicely that kind of protective effect is transferred via the microbiome, I suppose.
Yeah.
So this really shows that the effect on colitis of the Trichinella infection is at least partially driven by the gut microbiome. So it's not the Trichinella helminth on its own, but it's the changes that this is inducing, or at least partially. And that's kind of confirmed then in the next experiment. So
I am going to call foul on the next figure because they changed the colors.
They did, didn't they?
Yeah. So, which I found.
I thought I was going mad, but thank you.
Yeah, no. So figure 2, right? You've got the Bacteroides in this royal blue. Is that what it is? And the firmicutes are in orange, and then the others we won't. But then now you go to figure 4, and I'm like, wait, what's going on? But you know what they did? They changed the color. So now the firmicutes are royal blue. The Bacteroides are orange. So you're seeing the same effect, but they throw you with, and then you go to, panel D, and they're using the orange and the blue for, unclassified and, so. Use the same color. Be consistent.
You would have thought that maybe an editor would pick that up.
Yeah. Next time, send this. I'll review and I'll tell you. Use the same colors.
So, do you want to briefly summarize what's shown in that figure?
Basically, what they saw, if you don't get confused by the colors, is that, just being co-housed, you impact and change the relative abundance of the microbes. So, the T. spiralis changes the microbes in your, in your ones that are infected, but they also impact the co-housed folks.
Mm. And then they really start to kind of looking what is happening in the gut. So they looked at some metabolites produced in the gut, particularly short-chain fatty acids. And these are really produced when gut bacteria ferment fiber. And I think the most common types are butyrate, propionate, and acetate. And I think these short-chain fatty acids, they kind of have a role in supporting the intestinal epithelial cells, so the gut lining. So the results of that you can actually see in figure 5, and I just need to scroll to figure 5. I have to make it really big because, at my age, I don't see so well, so the scrolling takes a bit longer.
But it's a clear difference with these heat maps. I mean,
it's really nice. So it is clear that, the fatty acids produced in the Trichinella infected mice was really, really different from those produced in the control mice. And you can see that by the 2 different color kind of heat map things. I don't know, they look a bit like ellipsoids. So really different. So you can group them into there's essentially two different groups. So you got the group for the Trichinella infected mice, and you got a cluster for the non-infected mice. And in particular, what stands out is propionic acid, that really stands out maybe as a potential marker for Trichinella infected mice. So Trichinella infected mice, they seem to produce an awful lot more of that. And I suppose that does reflect a different microbiome in their gut. And I think it kind of confirms what we've seen in the previous experiment.
The question is, though, does it have anything to do with suppression of colitis, right? Suppression of inflammation, or is it just a byproduct of changing the microbiome?
I don't think they actually show that, don't they?
No, they haven't done any experiments. It would be nice to give mice propionic acid and see if that has any effect, right?
Yeah, I suppose that would be easy to do.
Connection here, like how does this connect unless you do something mechanistically like that?
Yeah. So I think maybe that's going to be in their next paper. Well, hopefully. Again, that might be something that could have been picked up.
Yeah, because I like figure 6, which we'll get into, starts to give what I would think, is a potential mechanism, right?
Yeah. So there's really there's 2 more experiments really, and these kind of then now finally demonstrate that the Trichinella infection changes the immune response and the also kind of structure or the kind of integrity of the epithelial barrier via, I suppose, a change in immune response. So first they kind of analyzed the production of IL-10, which is anti-inflammatory. And you can see that in figure 6. So here you can see that IL-10 is upregulated both in mice with colitis and Trichinella infection, but also mice with only colitis. However, there's a lot more IL-10 in Trichinella infected mice, demonstrating that this infection does increase somehow IL-10 in those mice. And the effect is then very much reduced in mice that were treated with antibiotic. And that kind of demonstrates that Trichinella-induced increase of IL-10 is really mediated by some members of the gut microbiome. And when the mice then were co-housed and the microbiome shared between them, there's also a significant increase in the IL-10 in the mice that only have colitis. And I think that is quite neat, really.
And the IL-10 is immunosuppressive. So
it is immunosuppressant,
That's part of the model then, that what's happening. Yeah.
Yeah, so IL-10 is going up, but then your bad inflammatory IL-1 beta, your IL-6 actually is reduced.
Yep.
So this to me, like mechanistically, plays into things that we know. And I like the fact that you can get this benefit by just hanging out with the T. spiralis infected mice, that you don't have to yourself be, because I don't want to be infected with T. spiralis.
But the question is, though, what's the mechanism of these changes in the ILs, right? When it's having to do with propionic acid, or is it something else, right?
Exactly. Yeah.
And also, you should do some experiments to give mice IL-10 and see if that ameliorates inflammation, right?
Yeah. Yeah.
Can you give IL-10?
Sure. You could do, and you could use IL-10 knockout mice.
Yeah.
And show that that it does. T. spiralis doesn't help at all anymore. Those would be interesting experiments.
Yeah, that would be good. Yeah. So I think the final experiments really looked at the kind of epithelial barrier properties during induced colitis. So they kind of looked at that by counting the goblet cells, which produce mucus and are kind of protective, so that I suppose it's the mucus that is protective of the epithelial cells, but, without the goblet cells, you won't have any mucus. And we can really see that colitis really decreases those goblet cells. And I think we've seen that in the very first figure as well, and essentially means that the epithelial barrier is damaged. And there are more goblet cells in Trichinella infected colitis mice. And then in co-housed mice, again, the goblet cell numbers between colitis mice with and without Trichinella is almost identical, kind of demonstrating the effect of the gut microbiome again. So I think that's all the kind of data that they're showing. So maybe just in summary, really, so they have kind of shown, although we don't know the mechanism, but they have shown that a prior Trichinella infection can reduce colitis disease severity in mice. And they also showed that this effect is no longer seen if the mice have no gut microbiome, i.e. if they were treated with antibiotics. And so essentially they have shown that the effect on colitis is mediated via the gut microbiome, which is somehow changed or affected by the Trichinella. And I think actually what I really liked is that microbiome and that kind of beneficial effect with having a pal in your cage. I thought that was really quite neat. I liked that also led to amelioration of the colitis symptoms.
Yeah, but humans live together. Some of them have colitis, some of them don't.
Yeah, no, that's true.
It doesn't seem to help them.
But they don't have Trichinella usually.
Yeah, I guess be careful who you're friends with, right?
Yes.
Whose hand you're shaking, Yeah.
Well, you need to be careful with that anyway in winter.
Yeah.
Did I miss anything, Daniel? Vincent?
No, I like, this sort of, for me, feeds into this whole of, okay, and I think we've covered similar papers where the parasites might have certain impacts, but the impact might be slightly indirect. I think maybe it was Dixon who talked about the parasites as like farmers cultivating their crops of microbes, and then the microbes are actually, really the player in this. So, because you're not transferring the Trichinella, but you're transferring the microbiota. It's transformed microbiota, and that seems to be having this protective effect. So, sort of this growing field of trying to modulate the gut microbiome for human health benefit.
So I mean, these are basically inflammatory diseases. So spiralis is suppressing inflammation. It's not surprising that you have a reduction in disease. It doesn't abolish it, right? It reduces it a bit. So
I think that's key. It reduces it a bit. It's not,
So the question is, how do we reduce that inflammation, right? So, you can give people microbiome transplants. So you could take probiotics, right? If you could identify the bacteria that are having this effect, you could give people probiotics. That's one approach. I'm sure people have tried immunosuppressive therapy for these inflammatory diseases.
Yes,
right?
yes, I think it's one of the management options.
It's management, but it's not eliminating it. So,
No.
I mean, if the microbiome has the potential to eliminate it, because if you can take the probiotic and reshape your microbiome, that could eliminate disease. Although I don't think this is going to eliminate it, right? I think it's going to reduce it, and there may be other factors as well.
Yeah, maybe it could be an additional form of therapy potentially for people who are maybe not responding to some of the more kind of traditional anti-inflammatory or biological treatment. I don't know. I'm wondering, where do we go from here? I mean, we're not, we can't really go out there and infect people with Trichinella. No. I mean, it's not the most harmful or dangerous pathogen, but I wouldn't really want to have it.
No, I don't think that's the cause.
The
cause here is to figure out
mechanisms,
how Trichinella does it.
Yeah.
And it sounds like it's microbiome. So now you have to focus on the microbiome. Can you identify individual bacteria and then give those to people? Or is there some other approach, right?
Yeah, I suppose there's going to be a lot of sequencing.
It's hard.
Experiments.
I mean, most of the gut bacteria are not culturable. That's the problem.
No, that's true.
So, it's hard to make a probiotics of something you can't grow.
There's a bit of correlation because they decide that, it's the bacteria. But, we were talking about previous paper. I forget maybe it was even on TWiP, Vincent, where we were talking about how there's the microbiome is not just bacteria. There's virus, there's fungi. Don't forget the archaea, which I always forget. Yeah. There's a whole complexity. And so what really is it that's being transferred, right? I mean, with antibiotics, we have a certain sense of, antibiotics, pretty broad spectrum, maybe not impacting the fungi, but maybe not impacting the viruses, but some of the bacteria have viruses, right?
Mm-hmm.
So when you were researching this, Christina, I'm sure they have done microbiome studies of people with these diseases. Is there any consistent change in the microbiome that they see in people with inflammatory bowel disease, for example?
I'm afraid I don't know.
Because I think we would know if there
I
were.
suppose so, yeah.
Right? But I think it's going to be different in every patient, and that makes it even harder. So.
I think, the microbiome is so different from person to person. It might be actually really quite challenging to draw any conclusions from any differences that you might see. It might just be just a genetic difference that you might observe in any case, or it would be really difficult to tease it apart, wouldn't it?
There could be multiple contributions, right?
Yeah, it would be really difficult to tease it apart. But I kind of like it that parasites are not just bad guys, really. Yeah.
Well, they're suppressing inflammation so that they can stay there, right?
Yes, I guess so. Yeah, it's in their own interest.
They're not looking after you. They're not looking after you. They're looking after themselves.
I guess so, but we can take advantage of that in one way or the other. Yeah.
I was looking at, going back to the quote unquote antibiotic treatment. And so what are these antibiotics? So it's ampicillin, okay, vancomycin. So ampicillin, right, is one of our penicillins. It's pretty broad. Vancomycin is pretty much a pure gram-positive agent. It's not absorbed. Neomycin, really broad. And then metronidazole, right? That's also going to affect some of the other, eukaryotic players in the gut. So, not just bacteria, not just anaerobes. So, it's pretty broad swath. So there may be more to the gut microbiota than just the bacteria that they focus on.
Sure.
Yeah.
Never simple.
But great stuff. I love this.
All right. It's TWiP 250. You'll find the show notes at microbe.tv/TWiP. You can send us questions, comments to TWiP@microbe.tv. If you enjoy these programs, we'd love to have your support. You can go over to microbe.tv/contribute to find out ways you can help us. Daniel Griffin is at Columbia University Irving Medical Center, parasiteswithoutborders.com. Thank you, Daniel.
Oh, thank you.
Christina Nell is at Glasgow University. She's also on Blue Sky, christina-nell.bsky.social. Thank you, Christina.
Well, thank you. And I do actually post our episodes in there, and every now and then someone picks it up. So, and I've noticed some of the other Microbe TV hosts are also there. You're there, Daniel? I'm going to look you up. I'll follow you.
We are there as Microbe TV. I didn't make a personal account, so Microbe TV is there.
Okay, I will look that up as well. So, amplify the message, really. Yep. So, it was fun.
I'm Vincent Racaniello. You can find me at microbe.tv. I'd like to thank the American Society for Microbiology for their support of TWiP and Ronald Jenkins for the music. So, Daniel, can you do the sign out today?
I'll try.
You've been listening to This Week in Parasitism. Thanks for joining us. We'll be back soon.
Another TWiP is parasitic.